Food intake is regulated by a variety of neurochemicals where some restrict food intake and some enhance it. Neuropeptide AF or NPAF and neuropeptide FF or NPFF are coded for by the same mRNA. NPFF has previously been shown to have an anorexic effect in chicks. We hypothesize that NPAF will have an anorexic effect in chicks as NPFF does. In this experiment, NPAF was given to chicks in increasing doses and food and water intake was measured. The presence of CFOS was also quantified in the arcuate, dorsomedial, magnocellular, paraventricular, ventromedial, and the lateral hypothalamic nuclei. Our results supported our hypothesis and demonstrated that NPAF causes anorexic effects associated with the hypothalamus. There are many complex processes that play a role in controlling hunger and satiety. There is still so much to learn and discover about this poorly understood process. These factors are defined by two qualities. Anorectic qualities decrease hunger and orexiogenic qualities increase hunger. One group of neurochemicals that are believed to play a major role in these mechanisms are the RF amides. Several of these chemicals have been shown to play important roles in these mechanisms while others remain poorly researched. This picture shows the general structure of an RF amide. Neuropeptide AF or NPAF is a member of the RF amide family, all of which have an arginine and amidated phenylalanine amino acid sequence on their carboxyl terminal ends. Neuropeptide AF is a part of the RF amide family. While it was initially discovered in cattle brains, it is also found in the brains of humans, rats, and mice. Some of the physiological effects of NPAF are somewhat understood, like its ability to stop the release of insulin and somatostatin from the pancreas, but its overall role in hunger and satiety is poorly understood, with little research being done. Neuropeptide FF is very similar to NPAF in that they both belong to the same chemical family and are synthesized by the same strand of precursor mRNA. There are many interesting questions about how this similarity might carry over into the function of the neuropeptide on the brain. There are several hypothalamic nuclei being studied in this experiment that play a role in hunger and satiety mechanisms. This diagram shows their relative positions in the hypothalamus. This experiment set out to explore the behavioral effects of neuropeptide AF as a function of hunger and satiety mechanisms. COB 500 chicks obtained the morning they hatched were used in this experiment. All chicks were kept in almost identical conditions. To prepare for injection, chicks were restrained and injected 3 mm anterior to the coronal suture, 1 mm lateral to the sagittal suture, and 2 mm deep aiming for the lateral ventricle. The injections were done freehand and the needles were left in for 5 seconds. The injection contained human NPAF, avian artificial cerebrospinal fluid, and 0.06% Evans blue dye. Once the experiment was completed, the chicks were decapitated and the presence of dye was verified in the lateral ventricle. If dye was not present in the correct regions of the brain, the chick was removed from the statistical analysis. First, both food and water were withheld from the chicks for 180 minutes. Chicks were then randomly assigned to 4 groups, each group receiving an injection containing either 0, 4, 8, or 16 nmol of NPAF. After injection, they had access to as much food and water as they wanted. Each chick's food and water intake was then observed every 30 minutes for a total of 180 minutes. Blood was taken immediately after the experiment and blood glucose was measured using a handheld one-touch basic glucose measurement system. ANOVA was used to analyze the data. When measuring water intake, the procedure was the same as in Experiment 1, but the chicks were food and water restricted for 180 minutes before the experiment and continued to be food restricted once the experiment began. When measuring blood glucose, the procedure was the same as in Experiment 1. Chicks fasted for 180 minutes were randomly assigned to groups receiving either 0 or 8 nmol NPAF injections and then allowed free access to food and water. After 30 minutes passed, chicks were anesthetized, decapitated, and the brain was fixed. The brain was sectioned, stained, and placed on a microscope slide. Light microscopy was used to view the slides, and the number of cells that reacted to the staining were counted. Data was analyzed using the ANOVA test. Chicks were fasted for 180 minutes before they were divided into 2 groups and injected with either 0 or 8 nmol NPAF. The chick's behavior was recorded and analyzed every 5 minutes. 30 minutes after the injection, food intake was measured. The behaviors quantified and analyzed were time spent standing, sitting, preening, perching, and in deep rest, as well as jumps, steps, feeding pecks, exploratory pecks, and escape attempts. The chicks in the 8 and 16 nmol groups displayed approximately the same decrease in food intake. Chicks in the 0 and 4 nmol group consumed about the same amount of feed. Chicks in the 16 nmol group had significantly reduced water intake from the 60-minute point on through the rest of the experiment. The 4.0 nmol group consumed significantly less water at minutes 90 and 120. Whole blood glucose concentration was measured at 180 minutes post-injection, and the concentrations are shown above. All concentrations showed a significant difference from the control, and glucose was dropped to about the same level for each of the non-control injections. The chicks injected with NPAF had significantly reduced water intake at all times during the experiment except the 30-minute mark. From 60 to 120 minutes, the 8 and 16 nmol groups had significantly lower water intake than the control and 4 nmol groups. At time 150 and 180, all NPAF groups had significantly lower water intake. NPAF did not affect blood glucose in food-restricted chicks. Blood glucose concentration in these chicks 180 minutes after injection is listed above. NPAF injections cause an increase in CFOS amino-reactive cells in the DM, PAMC, PAPC, and the VMH, but not in the ARC and LH areas of the hypothalamus. The behavioral effects of chicks injected with NPAF are listed above. Chicks injected with NPAF had fewer feeding pecks, less jumps, more deep rests, and less purging time. Exploratory pecking, escape attempts, defecations, distance moves, standing, sitting, and preening were not affected. NPAF was shown in these experiments to reduce food intake with a threshold for anorexiogenic effects occurring somewhere between the 4 and 8 nmol doses. This is shown by the changes in food intake from experiment 1. The 4 nmol group was unaffected while the 8 nmol group showed decreased food intake. These effects may continue longer than the 180-minute duration of these experiments. NPAF demonstrated a longer-lasting effect on food intake than NPFF, which lasts for up to 120 minutes post-injection. There are several other shorter neuropeptides that have varying length of their effects. This suggests that the shorter peptides like NPFF would have shorter-term effects on the brain and body than the longer peptides like NPAF, simply because one is longer than the other. Unlike previous studies with NPFF and NPVF, NPAF was shown in this study to affect thirst. Water intake was also decreased without the presence of food. This indicates that the water decrease was not related to a decrease in the water drinking that was associated with eating. This suggests that while there is some evolutionary conservation of traits in neuropeptides of the same chemical group, not all traits are conserved. This kind of complexity makes the mechanism of satiety and hunger difficult to study because the mechanism that leads to some traits like decreases food intake may be conserved, may not affect water intake, or may also decrease water intake. There is much left to be discovered in this field. Glucose concentration lowered with each injection regardless of the injection's effect on food intake. One explanation for this is that NPAF could stimulate the use of carbohydrates. Glucose concentration was not affected when food was restricted. This indicates that NPAF may play a role in glucose homeostasis. In experiments 1, the 4 nmol group supported the idea that NPAF plays a role in glucose homeostasis because while glucose levels decreased by 26%, the food intake remained stagnant. This differs from the 8 and 16 nmol groups who showed a decrease in both glucose levels and food intake. An increase in CFOS was shown in areas of the hypothalamus associated with the feeling of satiety, such as the dorsomedial, paraventricular, and ventromedial hypothalamic nuclei. This supports the idea that the effects of NPAF are caused by the perception of satiety. Analysis of the chick's behavior post-injection indicates that the decrease in consumption was not caused by an increase in anxiety levels. This is supported by the lack of behavioral change for behaviors like escape attempts. Many behaviors were observed in the experiment to determine which behaviors were affected by NPAF. There was no significant changes in behavior for the first 15 to 20 minutes. This suggests that there's a lag time between the injections and the behavioral effects. The behaviors that involve food intake, like food pecks, decreased. This doesn't show an inherent decrease in the amount of food consumed. To account for this, pecking efficiency was also measured. Pecking efficiency did not change after the NPAF injections. Overall, this experiment had an excellent design and was well executed. One possible change would be to have the experiment last for a longer amount of time. This would allow us to compare the effects of NPAF with other satiety-inducing neuropeptides in a much clearer way. We could learn the total time the reduction in food consumption lasts. Another addition to the study would be to perform the experiment on other animal models. Comparing the effects of NPAF on multiple species and for a longer time could both help with a practical application and maybe even the development of a drug for use in humans. The procedure of this experiment mentions that the chicks for each experiment came from the same hatch. It would be interesting to compare the results of an experiment of a similar nature to this one where chicks came from a variety of hatches to see if that has an effect on the results. It could also be interesting to do a separate statistical analysis of the chicks removed from the analysis when it was discovered that the injections weren't in the exact region of the brain that was desired. This would be an interesting separate analysis to see if the change in injection accuracy would vastly change the results of the experiment or if the results would still be statistically significant. This may answer some interesting questions about the ability of neuropeptides to move between regions of the brain. Once more research is done, this new knowledge could have many practical uses. If NPAF has the same effect in humans as it has in chicks, the development of a safer appetite suppressant with no anxiety-related side effects could be possible. This would, of course, require much more research as well as extensive clinical trials. If the drug is developed, it could be helpful for obese patients looking for an alternative to extreme exercise or surgery. This kind of information can also be used to understand the sheer complexity of the mechanisms that control hunger or satiety. This could then be used to create individualized weight loss or maintenance plans based on an individual's brain and body. It is really exciting to think that as a species we may be able to leave behind the uselessness of uninformed fad diets for individualized healthcare plans. Plans like these can also be used to help people or animals recover from nutrient-restricted situations more effectively by understanding the brain chemistry and neuroscience behind a healthy recovery while considering an individual's body condition.